Okay, good evening, all of you friends. Good evening. I'm extremely sorry for the inconvenience caused due to technical issue. I couldn't come on live at 7:30 p.m. So I'm just informing you prior for the next 1 week. I will be uploading all the videos through the recorded mode only. Okay, the video will be played exactly at 7:30 p.m. every day for the period of 1 week. So after 1 week, I will come to the live and all the lectures will be live only. I am requesting all of you for the period of 1 week, that is from this Saturday to the next Saturday. I am requesting all of you to watch this particular recorded classes only. So friends, in this particular crash course, I am going to cover more than 50 to 60 important topics, very important topics. I mean to say why I am using a word most important topics, because these are all the topics which were most oftenly repeated in the various entrance exams. So these topics which will be highly beneficial for the CSIR life science students, then followed by IIT JAM biotechnology students, then friends, followed by the most important for the GATE biotechnology also, as well as the GATE life science students also. Okay, so friends, I'm going to discuss each and every concept from basic to advanced level, from very basic to very advanced. I'll teach in such a way that after listening my lessons, you can understand the concept in very clear manner. Okay, so let's start with the first topic from the plant physiology, that is photosynthesis. All the classes will be held according to the schedule which was I shared before. Okay, so what are the various topics that we have to cover actually in the plant physiology? There are different topics are there: photosynthesis, respiration, photorespiration, nitrogen metabolism, plant hormones, sensory photobiology, solute transport and photoassimilate, secondary metabolites, then followed by the stress physiology. Friends, in this particular crash course I am going to cover the most important topics which will come in the exam for very sure. That is the photosynthesis, respiration and photorespiration, nitrogen metabolism, plant hormones, then friends, secondary metabolites. So let's start with the today's and most important topic, that is the photosynthesis. See, every CSR student, we can expect the question either in the part B or part C from this particular photosynthesis. And every IIT JAM biotechnology student, either for the 1 marker question or for the 2 marker question, compulsory we get the question from the photosynthesis. I can say one word: there is a no CSIR NET Life Sciences question paper, there is a no IIT JAM biotechnology question paper without the question from the topic photosynthesis. That this much important concept the photosynthesis is. Now everyone try to understand what I am talking now. So let's start with the photosynthesis. First I will explain you the concept, then I will make you solve the questions. By that the concept will be crystal clear to all of you. So friends, I am requesting all of you, as it is a recorded, I am requesting all of you, please kindly post your doubts, whether it's a small doubt or big doubt. I am happy to solve your doubts. Let's start. Photosynthesis. Friends, what is photo? Photo is light. Synthesis, to make something, to produce something. Friends, actually plants, what they will produce, we all commonly know that plant will produce their food. What is the food? The food is glucose. So how they can produce the glucose? By reducing the carbon into glucose. By reducing the carbon into glucose, they are going to prepare their own food. But friends, if the plant wants to produce their food, what they required? They require different type of raw materials. Plant require water, so plant will take the water from soil. Plant require carbon dioxide. Plant take the carbon dioxide from the atmosphere. Plant requires energy. From where it will take? It will take the energy from the sunlight. Never ever forget this particular point at all. And friends, here comes a very unique point. What was that? Whenever plant is going to reduce the carbon into glucose with the help of a chlorophyll. In other words, while preparing its food, plant is going to release oxygen as a byproduct. Plant is going to release oxygen as a waste product, as a end product. Always remember that, friends, none of the plant never ever release oxygen purposely, never ever. Every plant starts releasing oxygen as water, their waste product, their end product, their byproduct. That is what we exactly are breathing. And friends, we started reading this equation from 7th and 8th standard, a common equation you can remember. 6 molecule of carbon dioxide combined with the 6 molecule of water, which give rise to 1 molecule of glucose and releasing of the 6 molecule of oxygen. And friends, even though we are having availability of the carbon dioxide, availability of the water, what we majorly require? We majorly require light. We majorly require chlorophyll also. Please kindly do not forget that particular point at all. So with the help of a light and chlorophyll, plant is going to reduce the carbon dioxide into sugar. Now friends, in our competitive exam, most of the statements comes from where the particular reactions are happening. Not only for this, any chapter you may take, the most common statement related questions will appear. At least in a given 4 statement, at least 1 statement we can expect from where does this particular reactions are taking place. Now let's try to understand where does this photosynthesis occur. Friends, every one of you listen. Photosynthesis occur in a specialized organelle. What do we call it as? We call it as a chloroplast. Friends, if you look into the chloroplast, what's the common point you can understand? Chloroplast is a double membrane organelle. Yes or no? Yes. It consists of an outer membrane, it consists of an inner membrane. That's why we are calling it as a double membrane organelle. Friends, the space between the outer membrane and inner membrane, what do we call, my friends? Generally, we will be calling it as a intermembrane space. Do not forget that particular point. Friends, if you look into inside the chloroplast, we are able to see a flattened vesicle like structure. You are able to see. Friends, what do we call that particular flattened vesicle like structure? We call it as a thylakoid. Now the point is here, friends, do you think the thylakoid will be present as a single one? No. Every time, friends, thylakoids are present on one, like a group, like a stack. So friends, a stack of thylakoids or a group of thylakoids, what do we call it as? Friends, we will be calling it as granum. Never ever forget that particular point at all. So group of thylakoids are what we call granum. So friends, here comes a unique point. One group of thylakoids always connected to the another group of thylakoids by a thin thread-like structure. Friends, what do we call this particular thin thread-like structure? We call it as, friends, lamella. Do not forget that particular point. And what else the chloroplast consists of? You are able to see, friends, the chloroplast consists of ribosomes. We all know, right? What kind of ribosomes? 70s. And most of the students will get a confusion, or most of the students will get a doubt. What is the doubt that they will get? Originally chloroplast is present in the eukaryotic organism, so it should contain 80s. No. Friends, we read in the evolution chapter, according to the endosymbiotic theory, we all are evolved from what? Prokaryotic organism. What, friends? We are all evolved from the prokaryotic organism. That's why we consist of what? 70s ribosome. What else the chloroplast consists of? Chloroplast consists of a fluid filled structure. It's a cytoplasm, gel-like structure continuously moving within the chloroplast. What do we call that particular fluid filled structure? Friends, we will be calling it as what? A stroma, a fluid filled structure present in the chloroplast. So let me summarize whatever the points that we discussed so far. As I said that chloroplast consists of a outer membrane, inner membrane. The space between the outer and inner membrane of the chloroplast is called it as a intermembrane space, and inside the chloroplast flattened vesicle like structures, we call it as what? Thylakoids. Always remember. And group of thylakoids, we call it as granum. One group of thylakoids can be connected to the another group of the thylakoids by lamella only. A very simple point. And what else it consists of? It consists of 70s ribosomes. And I told you that the fluid filled structure present in the chloroplast, what do we call, friends? We call it as a stroma. What else it consists of? The very important, which was asked in the exam, if you look into the DNA of the chloroplast, friends, whether it is a chloroplast, whether it is a mitochondria, what is the common point that I want every one of you has to remember? Friends, the shape of the chloroplast DNA is the circular. What, friends? The shape of the chloroplast DNA is what? Extremely circular in nature. Please kindly do not forget that. And here comes a very unique point. What is the name of the protein that is coded by this particular chloroplast DNA? The name of the protein that is coded by this particular chloroplast DNA, we will be calling it as what, friends? Rubisco. We're gonna read in the dark reactions also anyway. Friends, why do we say Rubisco is the most important protein on the planet Earth? What is the reason behind that? Because, friends, 40% of every leaf of every plant present on the planet Earth, they contain Rubisco. That's why Rubisco is said to be most important protein on the planet Earth. Kindly do not forget that. What else the chloroplast consists of? You are able to see the large white color ones. What do we call them as? Starch granules. What's the function of starch granule? Which can store the starch. And you are able to see, my friends, yellow color ones. What do we call them as? Drop of lipids, which can store the lipids. I hope everyone understand the structure of chloroplast. Now, friends, we should also read a microscopic structure of chloroplast, that is the inner view of the chloroplast. Now you can ask me a question, sir, why we have to read the inner view of the chloroplast as we already read the entire structure of chloroplast? Because, friends, everyone listen my statements carefully. In photosynthesis, everyone listen, in photosynthesis, different reactions are taking place at different sites. Once again, understand, in photosynthesis, different reactions are taking place at different stages. That's why, or different locations. That's why, friends, we have to know each and every inner part of the chloroplast. Now I want every one of you focus on this. What are these, friends? Thylakoid, correct na? A flattened vesicle sac-like structure. Now you only try to understand my point. Here what you can see, group of thylakoids you can see, yes or no? Yes. My friends, what do you call this particular group of thylakoids? Friends, you call this particular group of thylakoids as granum. Correct now? What do we call it as what? Granum. Now everyone listen, what is the outer membrane of the thylakoid called? My friends, I am exclusively talking about the outer membrane. Kindly don't confuse, friends. The outer membrane of the thylakoid, we will be calling it as lamellae. Please kindly do not forget this particular point at all. The thylakoid membrane, what do we call, friends? Lamellae, outer membrane. And what about the inner space of the thylakoid? We called, friends, not only the chloroplast, you take any kind of organelle, you take any kind of organelle, the inner space of every single organelle, my friends, we generally call it as a what? Lumen. Lumen is the inner space. Please kindly do not forget that particular point at all. Now, let's try to understand what is this particular photosynthesis. Friends, whatever we are going to read in this entire concept of photosynthesis, I have summarized in this particular slide. If you understand this particular slide, the entire concept of photosynthesis will be clear to you. What exactly we have to read in this particular photosynthesis. I have already told to you, friends, the ultimate goal of photosynthesis is what? Reduction of the carbon, what? Reduction of the carbon into glucose. Yes or no? Yes. This is the ultimate goal of the photosynthesis. Yes or no? Of course, yes. Now friends, everyone keep it in your mind. Do you think plant can reduce the carbon into glucose that much easily? Do you think reduction of the carbon into glucose is a very easy process? No, friends, it's a very difficult process. Why? Because if the plant wants to reduce the carbon into glucose, it always require energy. What is energy? That is ATP and NADPH. So friends, first plant produce the energy, later plants can easily reduce the carbon into glucose. So don't confuse. If the plant wants to prepare the food, do not require energy. I'm so sorry. If the plant wants to reduce the carbon into glucose, do not require light. But if the plant wants to reduce the carbon into glucose, it require energy. Now the point is very clear. If the plants wants to produce the energy, what do they require? Friends, they required light. And for the production of food, they do not require light. That's why we call dark reaction. So friends, entire photosynthetic reactions are broadly categorized into two types. We will be calling it as light reactions and dark reaction. What is the difference between the light and dark reaction? Light reactions means it is always occur in the presence of light. My friends, what is the common point every one of you has to remember? What is the ultimate goal, ultimate aim of the light reaction? Why does the plant has to perform the light reaction? Friends, the plant has to perform the light reactions to produce ATP and NADPH. We all commonly know we read n number of times. ATP nothing but what? Energy equivalent. And my friends, what is NADPH? We all know that NADPH is the reducing equivalent. We all commonly know that. Then dark reaction, what I have told you, our ultimate goal is what? Reduction of the carbon into glucose. My friends, please kindly don't get confused. Dark reactions doesn't mean that they are occurring in the dark. Every student will get confused with it. Dark reactions doesn't mean that they are occurring in the dark. Dark reactions means that dark reaction means that they do not require light. They are light independent reaction. Now a simple question, most of the students will make a simple mistake. When does the light reactions occur? When does the light reactions occur? Day or night? Of course, our ultimate answer is what? Day. Because light reactions require the light, sunlight, so it occur only in the day. Dark reaction, most of the students will say that it will occur in the night. No, dark reaction doesn't mean that it is occurring in the dark. No, it means that they are occurring in the independent of light. Because in a simple common sense, plant has to prepare the food during the daytime also, plant has to prepare the food during the night time also. So friends, The dark reactions will occur actually during the day also, friends, even during the night also. Please kindly do not forget that particular point. Now, what exactly we gonna read in the photosynthesis? Briefly and elaboratively, we have to read this entire reactions. Which reactions? Light reactions. Then friends, followed by the dark reactions. Now you listen to me very carefully. When does this particular light reactions occur? When does the dark reactions occur? We understood very clearly. Now the point you people has to remember, where the light reactions are happening, or where the light reactions are taking place. Where is the site of light reactions? That's an important and unique point. Why? As I told you that, friends, in our competitive exam, majorly questions will be asked from where that particular reaction is actually taking place. Now let's start. I will explain everything briefly and elaboratively. Just focus. Light reactions, we can also call light dependent reaction. Now the first point you have to remember for the exam, where is the site of light reactions? Friends, if you quickly look into this picture, I have shown you, right, what I have said, this the outer membrane of thylakoid. What do we call the outer membrane of thylakoid? The outer membrane of thylakoid, my friends, we will be calling it as a lamellae. So lamellae is the site of light reaction. Thylakoid membrane or a thylakoid lamellae is the site of light reaction. Now you can ask me, so this is exactly the thylakoid membrane, plasma membrane. Now you can ask me a question, sir, why the site of light reactions is only thylakoid lamellae? Why it is not occurring in any other part of the chloroplast? That's a good question. The answer is that whatever the photosystems required for the photosynthesis, that is PS2, PS1, we are gonna read in the upcoming slide. Different type of electron acceptors like plastoquinone, cytochrome B6F, ferredoxin, they are all present where? In the thylakoid membrane only, thylakoid lamellae only. That's why, friends, we call the site of light reactions is thylakoid lamellae or the thylakoid membrane. Please kindly do not forget this particular point at all. Now my friends, every one of you listen to me very carefully. Photosynthetic, see, I have told you that the light reactions are taking place in the thylakoid membrane. Friends, if this particular reaction wants to occur, you have to understand, if a light reaction wants to occur, if the plant wants to produce ATP, if the plant wants to produce NADPH, first someone has to absorb the light. Yes or no? Of course yes. Who is going to absorb the light? Friends, I am going to provide you accurate and more amount of information which is required for the exam point of view. Hence I am requesting all of you not to skip even a single slide. Try to understand every single point. There are different kinds of photosynthetic pigments are there. Friends, all these photosynthetic pigments are broadly categorized into two types. One we called principal pigments, second one we called accessory pigments. Sometimes, my friends, the accessory pigments, we can also call it as antenna pigments. Kindly do not forget that particular point at all. Now, what are the principal pigments where all are present? So friends, chlorophyll A, bacteriochlorophyll A, chlorobium chlorophyll, these are all said to be a principal pigments. My friends, what is the difference between the chlorophyll A, chlorophyll B, C, D? Friends, don't think that every green plant or every higher plant, I can say, they do not consist of all kinds of chlorophyll pigments, which is absolutely a wrong statement. What is the point you have to remember? Higher green plants, they majorly consist of chlorophyll A, and friends, then most another important one, bacteriochlorophyll A, which is present in the purple bacteria. Now you can ask me a question, sir, what is the difference? What is the difference between the chlorophyll A and bacteriochlorophyll A? You can ask me a difference. Then I can tell you, my friends, the difference between the chlorophyll A and bacteriochlorophyll A is in terms of a structure. Listen, a modified chlorophyll, listen friends, modified chlorophyll present in the purple bacteria, which we call it as bacteriochlorophyll. Once again listen, a modified chlorophyll present in the purple bacteria, which we call bacteriochlorophyll. Next, the third one, modified chlorophyll present in the green sulfur bacteria, which we call chlorobium chlorophyll. My friends, please kindly don't get confused. All are chlorophylls only. Sir, what is the difference? They are differ in terms of a structure. I'm going to explain you in the next slide. What they are differ? They are differ in the structures especially. Now, antenna or accessory pigments. Now you keep it in your mind. What are antenna or accessory pigments? I told you, right? Chlorophyll B, C, D. Friends, do you think chlorophyll B, C, D, are they present in the higher plants? No. Friends, chlorophyll B, C, D will be present in the lower organisms. Especially they are present in the green algae. My friends, I want you people to remember this for the exam point of view. Which pigments are present in which area? Now here comes a very important. Carotenoids. Friends, where this particular carotenoids are present? Friends, carotenoids are present in the most of the plants also. Even certain bacteria also consist of this particular carotenoids. The carotenoids are of basically two type of friends. What are the two type of carotenoids? Everyone please try to understand what I am talking. Carotenoids are of two types. One is carotins. Another one is what, my friends? Xanthophyll. Now, if you ask me a difference between the carotin and xanthophyll, what I can say in terms of a structure? I can say, my friends, carotins are actually consist of just carbon, hydrogen, nothing else, friends, nothing else. That is what the carotin contains. But if you look into the xanthophyll, what's the common point you have to remember? Along with the carbon, hydrogen, my friends, it also consists of a oxygen. That is what? Xanthophyll. Friends, what are the best examples of the carotene, which you have to remember for the exam, which was asked in the exam? The best example of the carotenes is what? Beta carotene. Example, where the beta carotene is present, we all commonly know that present in the carrot. I hope every one of you remember there is another type of carotene is there, my friends. We call it as a lycopene. Friends, where the lycopene is present, tomato. Tomato is in red in color because of what? Of course, because of the lycopene only. What I have told you about the carotenes, they just contains carbon and hydrogen. What about the xanthophyll? They consist of carbon, hydrogen. In addition, it also consists of a oxygen. My friends, what are the examples of xanthophyll? Every one of you keep it in your mind. The best example of the xanthophyll is, friends, lutein. Lutein is the best example of this particular xanthophyll. Do not forget that particular point. Now, phycobilins. Where this particular phycobilins are present? They are present in the cyanobacteria, rhodophyta. Friends, whichever the information I am covering, once again I am saying to all of you, everything is an important and unique information. Try to remember every point. They are present majorly in the cyanobacteria and rhodophyta. You people try to remember the example. Phycoerythrin, phycocyanin and yellow phycocyanin. Please kindly do not forget these points at all. Friends, Right now, whatever we studied is different type of pigments that are present in the plant, present in the algae, present in the rhodophyta, present in the bacteria. Now, my friends, let's try to understand the structures briefly and elaboratively. What are the structures? See, everyone keep it in your mind. You no need to remember the structure of the chlorophyll, structure of the carotenoid, structure of the bilin pigment. But what you have to remember, friends, you have to remember the structural composition. How does the particular structure looks like? What does that particular structure is composed of? What does it made up of? Those things we people has to remember without a fail. Okay, now let's try to understand, my friends, very clearly. If you look into the structure of a chlorophyll, my friends, everyone understand. This is the structure of chlorophyll. I'll explain elaboratively, focus. This is called head of the chlorophyll. This is called tail of the chlorophyll. My friends, this is head, this is tail. Friends, the head of the chlorophyll we called porphyrin head. Friends, what do we call the tail of the chlorophyll? My friends, the tail of the chlorophyll, we will be calling it as a phytol chain. Please kindly do not forget this particular point at all. Okay, now let me define you. What is porphyrin head? How does it look like? I want every one of you pay attention here. If you look into the structure, you are able to see A, B, C, D. What are those? They are pyrrole, 5 carbon containing compounds. What, my friends? Pyrrole, 5 carbon containing compounds. Listen everyone carefully. So total how many pyrroles are there? 4 pyrroles. Friends, 4 pyrroles we call tetrapyrrole. A tetrapyrrole ring, listen, a tetrapyrrole ring come close together and surround the central metal atom magnesium with the help of a nitrogen linkage. I am telling to all of you once again, listen to me carefully. What is porphyrin? A tetrapyrrole come close together, surround the central metal atom magnesium with the help of a nitrogen link. That is what exactly we call, friends, porphyrin ring. Now, once again, most of the students will forget what is the function of magnesium. Many times asked in the exam also. See, I was continuously and repeatedly saying that chlorophyll is going to absorb the light. Yes or no? Of course, yes. My friends, how does the chlorophyll is going to absorb the light? With the help of a magnesium only. So, my friends, what's the major function of the magnesium here? Friends, magnesium is basically acting like a chromophore. Never forget this point, friends. What's the function of a magnesium? Magnesium acting like a chromophore, chromo color 4, which can absorb the color. That is porphyrin head. And what is phytol chain? Phytol chain is a very simple, my friends. It consists of a long hydrocarbon. Simple long hydrocarbon chain. Now, let me tell you one unique point, friends, here. What was that unique point? As I was continuously saying that in the plant and in certain organism, chlorophyll A is there, B is there, C is there, D is there, bacteriochlorophyll A is there, different type of chlorophylls are there. But friends, what is the common point here? Every single chlorophyll having head and having tail in common. Now, my friends, how can we differentiate one chlorophyll molecule? Listen my statements, friends. How can we differentiate one chlorophyll molecule with the another chlorophyll molecule? My friends, here is the answer. It is exclusively based on the side chain. What, my friends? It is exclusively based on the side chain. Let me explain you how. You don't need to remember all this, but you have to understand how we are differentiating one chlorophyll with another chlorophyll. That is important for exam. So now you try to understand chlorophyll A. It is having a side chain methyl group. But my friends, if you look into the chlorophyll B, it do not have a methyl group. What does it is having? It is having aldehyde group. That's how you can differentiate. Now if you look into the bacteriochlorophyll, it is having one more. So friends, what's the point you have to remember here? One chlorophyll molecule can be easily differentiated from the another chlorophyll molecule exclusively based on their side chain. This is about the structure of a chlorophyll molecule. Now my friends, let's try to understand the structure of the carotenoid. Do not forget that. Friends, if you look into the structure of a carotenoid, what is the common point I want you people to remember here without fail? Everyone listen to me. Carotenoid, my friends, I will be calling it as isoprene. Now you can ask me, sir, what exactly isoprene mean here? My friends, once again I'm telling to all of you, you no need to remember the structure. You have to remember is the structural composition. Isoprene nothing but friends, 5 carbon containing compound. What my friends? 5 carbon containing compound with the alternate double bond, single bond, double bond, single bond, double bond, single bond. So a 5 carbon containing compound with an alternate double bond, single bond, that structure only we will be calling it as a isoprene structure. What is the best example? The best example is the beta carotene. Please kindly do not forget that particular point at all. Now next, bilin pigments. Friends, you are able to see, don't confuse, here comes an interesting point. Bilin pigments also consist of a 4 pyrrole, 1 2 3 4. So we call it as a, undoubtedly, what do we call my friends? We call it as a tetrapyrrole, no doubt about it. My friends, just now where we heard about the tetrapyrrole, if you quickly recall my words, just now we heard about the tetrapyrrole here, where? In the chlorophyll. But my friends, what's the unique point you people has to understand here? Here the tetrapyrrole come close together. Yes or no? Yes. But here what you have to remember, friends, here you have to remember it is a linear tetrapyrrole. That's the difference. 1 2 3 4, which are linear, straight chain. So friends, linear tetrapyrrole is a bilin pigment, isoprene structure is a carotenoid, porphyrin and phytol chain, that is what? Chlorophyll. I hope my friends I made the structures very clear to all of you. Now one more interesting point. We all know that all these pigments are responsible for what? Absorption of light. Now let me ask you a very simple question. If chlorophyll, carotenoid, bilin pigment all are absorbing the light, then friends, why the plant need all the pigments? Any one is sufficient, right? If plant is absorbing, if the pigment, all pigments are absorbing the light, any one is sufficient for the plant, then why plant require many? The reason behind that, different pigments absorb the light at different wavelengths. Please kindly do not forget that particular point at all. Different pigments absorb the light at different wavelengths. In addition, let me tell you one interesting and unique point about the carotenoid. What was that? Of course, carotenoid is a photosynthetic pigment, absorb the light, no doubt about it. And friends, it is going to protect the plant body from where? It is going to protect the plant body from photooxidative damage. What is that? Photooxidative damage, nothing but high intensity of sunlight. You try to understand, we are human beings. When we exposure to the high intensity of sunlight, we will try to escape. We will try to escape. Yes or no? Of course we will try to escape. Do you think plants can escape? Plants can move, plants cannot move, so they have to tolerate it. So how they can tolerate? With the help of this particular carotenoids only. So carotenoids absorb the light also. At the same time, friends, they can protect the plant from high intensity of sunlight also. Now, as we understand that the chlorophylls are going to chlorophyll, carotenoid, bilin pigments absorbing the light. Now, friends, based on, listen, it's a very important, based on the absorption of light. This slide is a very important slide, my friends. I'll try to make it simple. Focus, everyone. Based on the absorption of light, there were two spectra were derived. What, my friends? Based on the absorption of light, two spectra were derived. What are those? Absorption spectra, then friends, followed by the action spectra. I will explain in a simple way. Focus. Friends, listen my statements, what I am talking. Absorption spectra, nothing but at which particular wavelength, what friends? At which particular wavelength the particular pigment absorbs the light. Once again, listen. At which particular wavelength the particular pigment absorbs the light. That is what explained by the absorption spectra. Please kindly do not forget that point. Now every one of you look into the given table, given graph. Here on the x-axis we have taken wavelength in nanometers. On the y-axis we have taken the amount of light absorbed. So friends, every one of you keep it in your mind. If you look into the absorption spectra, listen, chlorophyll A was given, chlorophyll B was given, carotenoid also given. Now everyone look into the graph. Chlorophyll A absorb the light, see here, at a dark blue light, light blue light. But you see, my friends, the peak was completely down. It cannot absorb the green light. Now every one of you remember the statement what I am saying right now. Any pigment, whatever the color it is absorbing, it never ever appear in that particular color. Whatever the color it is reflecting, it will only appear in that particular color. Never ever forget that statement. Once again I am saying you, whatever the color absorbed by the pigment, it never appear in that color. Whatever the color reflected by the pigment, it will always appear in that color. Here, my friends, chlorophyll A absorbed which color? Dark blue color, light blue color. But do you think the chlorophyll A absorbed the green light? No. Instead of absorbing the green light, it reflected the green light. What, my friends? It reflects the green light. Since it reflected the green light, the plant appears to be green in color. Now you take another example. You take the yellow color carotenoid. See here, the graph is very high, up to 530, 540 nanometer, which means it can absorb, carotenoid absorb dark blue color, light blue color, green color. But my friends, it cannot absorb yellow color, cannot absorb red color, cannot absorb orange color. Where the carotenoid is present? Carotenoid is present in the carrot. So the carotenoid which is present in the carrot reflect the red light. So it appears, reflect the orange light. So carrot appears in the orange color. You take example of tomato. Tomato also contains a carotenoid. I have told to you. What is the name of that? Lycopene. Lycopene is reflect what color? Red color. That's why tomato appears to be red in color. So my friends, what's the summary? Whatever the color absorbed by the pigment, it never appear in that color. Whatever the color reflected by the pigment, it always appear in that particular color. That is what do we call absorption spectra. Right. So next one is what? Action spectra. Friends, this particular action spectra we will discuss in the tomorrow class. So tomorrow, along with the light reactions, I am going to finish the dark reactions also. Please kindly don't forget. Please, and my sincere request to all of you, till next Saturday, till next Saturday all the videos will be released at 7:30 p.m. sharp every day, which will be completely recorded. The live is going to be start from the next Sunday 7:30 to 8:30 p.m. Okay, so friends, I am requesting all of you watch the complete lecture, and my sincere request to all of you, whatever the doubts you have, so please kindly post your doubts in the form of a comments. 100% I will give the reply, and I'm always happy to help you in solving your doubts. Okay, thank you all of you friends. I'll meet you in the tomorrow 7:30 p.m. sharp. Thank you all.